All right, recording. Okay, so you can give it a little stir when you're getting the solution just to make sure all the sugar is dissolved in there. We have 50%, 25%, 10%, and then we have plain water, which is going to serve as our control, which we'll go over in a moment. As I said before, we have yeast. This is just the regular kind of yeast that you use to make bread, which if you've never made bread, I recommend. It's pretty fun. Then we also have some stuff to label our tubes so we know everything that's going on. Each lab station is going to have 4 test tubes, 4 balloons, their own yeast, a measuring spoon, a graduated cylinder, a ruler, and that's it. All right. So the other thing that's going on is I brought in a razor to shave my onion, and I actually ended up using my finger towards the end, but I made onion slides. I don't know if you guys can see that. There's a tiny little thin piece of onion on there, also a hair, and we're going to be looking at the onion cells under the microscope. I just did a test run on one of the slides. It looks really awesome. And then we have a salt water solution. All I did was just take table salt and put it in some water. We're going to be putting that on the onion cells to see what happens to them. And then otherwise, we're going to be talking about cellular respiration, talking about cells a little bit, reviewing that content, right? I know cellular respiration is a really difficult concept because it has like 4,000 vocab words in it. So we're just going to we can all just gather around here. You guys can get a stool if you want. We're going to look at the lab document itself. So if you have, oh good, you've done this before, cool. So hey, you're here, Samantha. We're just opening the lab document and we're actually just going to go through the instructions a little bit. And we're going to talk about the background and the content. So I'm actually going to share my screen with you guys at home so I can open the lab document. Here we are. So part 1 is going to be the onion cells with the microscope, right? We only have one microscope, so we're not going to be able to necessarily, all do that at the same time. So we're kind of just going to go over there when it's free, do that part, come back over to do the yeast. Right? Because we're going to have to be waiting 5, 10, 15, 20 minutes for the yeast anyway. So while you're waiting around, microscopes. But basically these circles that are on the screen or on your paper, right, that's where you're going to draw what you see. And in theory, we should be able to see a cell wall, a cell membrane, and a nucleus. The nucleus is a little hard to see. I could see 2 of them on the sample that I prepared. When we add the salt water, it might be a little easier to see. So you're going to predict what will happen to the onion cells if we add salt water. A lot of you guys already know the answer to that question, which is fine. Write it out. Use a science word, right? Describe why that's going to happen. And then we're going to draw what happens after we add the salt water. So we'll probably all look at the same sample, add the salt water, and then all look at it. That way we don't have to keep switching it. And I don't have to shave the onion anymore. Because then now my hands will smell like onion for 3 days. Okay. So then you're going to explain, right? Why did the onion change? Again, using science words. As for cellular respiration, hopefully you guys were able to read through some of this text in advance. But right now we're going to talk about it briefly here, right? So cellular respiration, the main purpose of cellular respiration is to produce ATP, to produce the energy that powers your cells, right? The problem with cellular respiration is that it's really complicated and has like 50,000 steps, right? Which here they emphasize that makes it more efficient, which is true, but it also leaves room for problems, right? So if we look at our formula here, we're going to be taking glucose and oxygen. When we have those 2 things, we produce carbon dioxide, water, and energy. If we're talking about in a human, what happens to that carbon dioxide in the human? You breathe it. It comes out of you. It's toxic, right? If you hold all that carbon dioxide inside of you, you will die. You breathe it out, right? What happens to that water? You pee it out. It's waste. It leaves your body, right? And then what happens to the energy? You use it, right? Really simple concept here. These are 3 things that you're every day doing, right? You're breathing, you're peeing, and you're using energy. So the thing that is going to happen with our yeast, though, if we're putting a balloon on top of a test tube here, are the yeast going to be able to use oxygen?
No.
No, right? Whatever oxygen was in the tube, they'll use, and then they're going to run out. Well, guess what? They can still make energy, right? Because yeast don't want to die if all of a sudden there's no oxygen. So they will go through what we call fermentation, right? Or anaerobic respiration, where there's no oxygen present. So humans, because we're smart, we have decided, wow, that's a cool thing. We can use that to make beer, to make wine, to make bread, right? We can make all of these products to make cheese using yeast and taking away their oxygen, right? So if you think about beer and wine, right, that's like a huge thing in like the Portland area, right? In fact, lots of my friends go work on farms and pick grapes and make wine. They just have these huge barrels where they put all the ingredients, they seal it up, right? And they just leave it. And what's happening is that yeast is not having oxygen. It's going through anaerobic respiration. It's producing alcohol as a byproduct, right? And then the yeast die eventually, but we don't feel bad for them because we're happy that we produced alcohol, right? So it's fine. Regardless, the same thing can happen to you, right? If you've ever been exercising vigorously and you get a cramp, right? You're running and then, what happens? Are you running through the cramp? No, you're like, you have to stop. That is a buildup of lactic acid in your cells. That means that those particular muscle cells ran out of oxygen. They start producing your version of alcohol, which is lactic acid, not as nice as real alcohol, right? And it actually will kill your cells if it's not gotten rid of immediately. So humans, we can't live without oxygen, right? Parts of your muscle can live for a few moments without oxygen, but then as soon as that starts hurting, you stop, right? Because your muscles need to get oxygen. That's also why you breathe faster when you're exercising. You want to get more oxygen into those muscles, right? So it's all related to this process. I put this diagram here. Sorry, people at home, I'm not even on the right page on the virtual thing here. I put this diagram that has like a really good summary of cellular respiration. Really, anytime you're going to be tested on this, you can use your notes, you can use the diagrams, right? So it's not so important to memorize this, but it's to understand that there's 3 main parts. There's glycolysis, right, which is the initial step. That's where your body says, okay, is there oxygen here? There's oxygen. Yes, we're going to make tons of energy. Everybody's happy, right? If there's no oxygen, you're going to make that lactic acid, that waste product, and you're only going to produce 2 ATP. Usually you produce 36, right? Would you rather have $36 or $2?
36.
36, right? Guess what? That $2 is only enough for you to go through the process one more time and hope that there's oxygen, right? And if there's not, you still just have $2 and your body will keep trying, right? And then if there is no oxygen, you die, right? Some organisms, like bacteria, they can actually exist without oxygen, right? That's possible for them. This fermentation process, they can actually utilize that in a positive way. Yeast can exist for a pretty long time fermenting. It is enough energy for them to keep going because they're really small, right? A yeast is a cell. It's easy to power a cell with 2 ATP. It's hard to power a body with 2 ATP. So yeast can exist in both, right? They can have oxygen or not. Eventually they will die, right? They can't live forever without oxygen, but that's part of why they kind of grow everywhere because they can pretty much grow in any condition. So on page 3, there is this little diagram that I would like for us to fill out together so that we can be thinking about the difference between these 2 processes. All right, so they have this circle here. Some of these are like, kind of like, what do I put there? But then once you know what the answer is, you're gonna be like, oh, that. So I believe in this first circle, people at home or people here, correct me if you think I'm wrong, but I think it's glucose, right? We start with a glucose. It goes through glycolysis. We get something called pyruvate, right, which is just a broken apart glucose. And then we go and produce carbon dioxide, and we produce 2 ATP, right? Well, we also produce water. I guess they want water there. So put water here. How do I do this? There we go. Water. And then we only produce 2 ATP. That's supposed to go in the little box, but it didn't fit. Okay. Oh, wait, no, this is with oxygen. What am I saying here, guys? No one's correcting me? 2, we produce 36 ATP. I thought this was without oxygen, sorry. I wasn't reading my own thing. How do I delete this? Go away, 2. I don't want you. Uh-oh, it doesn't like me. Okay. Oh, wait, here we go. Maybe I have to, whatever. 36. Okay, 2. How do I get rid of the 2, though? There, no. It's not going away. Okay.
Highlight it and then delete it.
Okay, I deleted both of them, but I got it away. 36 ATP, right? Sometimes 34, 36 will go. Okay, without oxygen, right? We have 2 different versions here. We have the muscle cells, right? That's you. And then we have plants and microorganisms, which would be yeast, bacteria, etc. So, you can't hear anything. Is that true for everyone else? Turn your volume up. Okay, everybody else can hear. Okay. Try turning your volume up, and then you can't type in your circle. Yeah, sorry. I'm using the annotate thing, so I can type in the circle. You can just write underneath it. That's totally fine. It's a visual diagram. Why can't I move this chat box? Okay. Let's go away thing. All right, sorry guys. Got a lot to manage here. Okay, chat. Okay, there we go. Moving the chat. Okay. So without oxygen, the muscle cells, right, we start with the glucose again. They go through glycolysis. And then, I wonder what they want us to put in the circle. We could go there. Uh-oh, somebody's needs to mute themselves. Okay. All right, so the glucose goes through glycolysis, right? And it gets broken down. I think pyruvate goes there again, right? It gets broken down into pyruvate. And then it doesn't go through the Krebs cycle and the electron transport chain, right? It just produces lactic acid.
Where that line is?
Yeah, I think that line is supposed to just tell you that it goes through the process in I think it's still called fermentation in humans as well. You just don't produce alcohol, you produce, yeah, I think where the line is, they want you to write fermentation, because that's what the process is called. And then from fermentation, you produce lactic acid. Okay.
And the ATP produced?
We'll just say you produce lactate, because that's what they wrote here, and that probably is the correct way to put it. Makes lactate. Right, and then how many ATP are produced? Now we only have 2 ATP. Okay. So, oh geez, this text box is like going crazy over here. All right. So pyruvate gets made still, right? But now instead of going through the Krebs cycle, going through the electron transport chain, which really is just molecules passing in and out of a membrane to produce ATP, right? Instead of that happening, we ferment and we make lactic acid, right? Or lactate. And that's the burning, unpleasant feeling that you feel in your muscles when they are lacking oxygen, right? Plants and microorganisms, it's going to be the same stuff over here, except that instead of lactate, they make alcohol. So you also still make carbon dioxide, right? And you still make water. But really the issue here is that you don't make a lot of ATP. What's going on with this thing? All right. Yes, so I'm not doing anything on the actual doc. I'm just doing it on the Zoom. That is a good point from Jacob. You're doing it on your own document. I know you can't really write in the circles on the computer, but just write underneath it. All I want you to get out of this, with oxygen, lots of ATP. Without oxygen, not very much ATP, right? The details are not as important. And also, I'm not sure why they have 2 circles here when they only had 1 for the muscle cells, because it should be the same minus the alcohol, but we'll see. So that is really just to get our minds around what we're doing. Okay, so in this lab, right, you guys are like, all right, cellular respiration, we got it. What we're going to be doing is using this process of cellular respiration, we're going to be answering the question, does the amount of sugar or glucose impact the speed and the amount of product produced? Right, so as the yeast are breaking down the sugar, what are they going to be producing? They're going to be producing alcohol, right, which we'll smell it after and see if we can smell it. What else are they going to be producing? What's going to happen to the balloon? It's going to get bigger because it's getting filled with what? Carbon dioxide. Oxygen is a good guess because that's the only other gas pretty much that we talk about. Carbon dioxide, right? This balloon is going to get filled with carbon dioxide. So if the sugar impacts this, right? Let's say that a lot of sugar, if the yeast goes faster, then we would expect that the balloon is going to get bigger, right? And it's going to get bigger faster. So at 5 minutes, it's going to be bigger than the other ones. And then at 20 minutes, it's going to be huge, right? So we would expect that it would happen faster and it would get bigger. If you think it doesn't matter, right? The amount of sugar doesn't matter. The yeasts are just going to process it as fast as they can. Then all of our balloons should be the same, right? That's a perfectly valid hypothesis as well. You could also argue that less sugar, the yeast will go faster, right? I don't know exactly what your argument for that one would be, but you could argue that.
Okay.
So you guys are going to be writing a hypothesis in a second, right? If you already have one in mind, you can start jotting it down, but you're going to be saying what amount of sugar is going to produce the biggest balloon the fastest, right? And our percentages, again, are we're using 50% sugar, 25% sugar, and 10% sugar. And that's mixed with water.
Okay.
Unless it pops, yes. If the balloon pops, then we'll assume that yeast was really fermenting. So is this going to be a big grade? Nope, it's just going to be like a 40-point lab. Oh yeah, actually, I'll talk about that now. So Canvas, because it hates me, you can't add new assignments to Canvas once the course has started, which is why all the labs that I've added before have gotten deleted. You may have noticed, like if you were trying to submit whose doorknob is the dirtiest, it was gone, right? Because like every time the due date came, it kept just deleting it. And they don't know how to fix that. It shouldn't be that hard, but they, for some reason, think it's hard. So for now, you're going to be emailing me the labs, and I'm going to keep track on a spreadsheet of how many labs you do. And then at the end of the semester, I'm going to add points to your grade, right? So if you do no labs, it's going to hurt your grade. If you do all the labs, you're going to end up getting extra points. So that's how we're going to be kind of tracking this, which is annoying, and hopefully they fix it. The other option is that I create another class just for labs, which I thought about doing. But then you guys are going to have more classes, and it's going to be a little bit more to organize. So I'm kind of still deciding if I want to do that. Okay. variables. This,
Is it the items that we're going to be using?
yes. So variables, okay, goodbye, cellular respiration stuff. All right, variables, I will actually type in the document now for those of you in the document. So we have our independent variable, our dependent variable, our control variables, and in this case, we actually also have a control group. So we have a lot of variables here. So what are we actually testing here? What did we change about this experiment? Think about the question, how does the amount of sugar impact the speed of cellular respiration? Exactly, the amount of sugar. So we're changing the amount of sugar. Specifically, we're doing 10%, 25%, and 50%. What are we measuring? We're looking at time, and what else are we looking at?
The balloon?
The balloon, right? So in theory, we would measure with like a measuring tape so we could get the circumference, right? But we don't have that, so we have rulers. Carbon dioxide build is an appropriate dependent variable as well. You could just say the amount of carbon dioxide. So we'll have options here. You can measure the diameter of the balloon, right? Or I might have a piece of string where you could put it around the balloon and then lay it on the ruler and see how long it is. But we're probably going to do diameter. So for a dependent variable, we're going to do time and diameter of balloon, right? Circumference is ideal, but we might not be able to do that. And time, really, we're just going to be looking at speed here, right? All right, control variables, things that we're keeping the same here, right? We're all using glass test tubes. We're all using balloons from the same package. We're all using the same kind of yeast, right? From the same, they were all in like a strip, so assuming they're from like the same yeast population. So same yeast, same test tubes, same balloons. There is one group who will have one tiny test tube and 3 huge ones, but shouldn't be too big of a deal. Well, I spelled balloons really weird. All right. And then our control group. So here's the thing. Let's say that the yeast were just producing carbon dioxide no matter what, right? How would we know that? Like, what could we use here to sort of compare? We're mixing them with sugar water, and they're going to produce gas. So what could we mix them with to know for sure that it's because of the sugar that they're actually doing something?
Good question.
It is a good question, right? Thank you, Giovanna. Water. And Jacob? Yep, just plain water, right? So we're going to have one test tube that just has plain water in it, still has the balloon, right? And in theory, it shouldn't grow at all, the balloon, right? If that balloon grows a lot, then there might be something wrong with our experiment, right? There might be something wrong. Maybe the test tube's dirty, the balloons are weird. I don't know. We'll figure that out if it happens. Hopefully that doesn't happen. Okay. So hypothesis, you're going to write really quickly before we start the experiment. In terms of procedure, I changed the amount of sugar solution just because the test tubes are pretty small and 25 milliliters would not fit in them, so I changed it to 10. You can still use a 1/4 teaspoon of the yeast. If you would like to use like half of that and try to do an 1/8, you can. When you're shaking the tube, right, you're going to put in your solution, put in your yeast. Might be a little hard to get it in the tininess. Actually, I have a funnel. We'll get the funnel out. You're going to put the balloon on, and then you're just going to gently shake the tube. Don't go like this, right? We don't want it in the balloon. You can even cap your finger on top of the balloon and shake it a little bit just so the yeast dissolves, right? And then once it's dissolved, you're just going to set it in the rack, and we're going to leave it. Such a fun noise. Okay. And then we're going to be recording here after 5 minutes, we're going to record the diameter of the balloons. After 10 minutes, after 15 minutes, and then after 20 minutes. And then we'll move on to the analysis questions, which we might not have time to do here, but maybe. Other things, next week on Tuesday is an optional lab. It's just going to be like a work time. So if you have specific questions, you don't understand like photosynthesis and you want me to walk you through that, then come in and we can sit together and talk about that. I'm also going to have physical science people coming in, and I will be giving extra credit for people who come in and have a real question. Like if you just come in and sit there, like, I'm not giving you extra credit. But if you come in and you're like, hey, can you help me with this question? Can we look at this assignment? Then I will be giving extra credit. How many points? Probably 10 points. And we'll add it to whatever assignment you want right there on the spot. Okay, so that's going to be a great time. It's like about halfway through the semester, right? So look at your grade, look at what you've submitted so far, see if there's any assignments that you're just not understanding, right? That's why I'm here, and sometimes it's nice to sit next to each other and talk through something. Okay, that was a lot of talking, but we're done with that. And can you put those points on an exam? You can. You can put them on anything. All right, so.
Do you want two of us to?
Yes.
Sweet.
So we're going to have some lab partners here. Ooh, we have 18 people.
That's a lot.
19, 20, 21, 22, 23. Yes. Okay. I'm excited. I want to have the most people attend my virtual sessions, guys. It's a goal, so always come to them. Okay. Oh, other thing, a poll. Very quickly. If I made a biology Snapchat, would that be something that you guys would like?
Yes.
That'd be cool.
Okay. Because I was thinking about it because I love Snapchat. Zach says no. Okay, thanks. That's fine. You don't have to follow it. But I was thinking I could post really fun science facts, right? And I won't follow you. I don't have to like see your Snapchat. You could just follow mine.
Sweet.
Right.
Well,
Although
yeah.
I do follow my students from New York that I moved away from. I see some really strange stuff on there. All right. So yeah, it would just be my thing that you could add. I won't follow you on Snapchat, so I don't see whatever you're doing. Okay. And if you don't have one, then if you ever get one, biology theme. Okay. So something I was thinking about. All right. We're going to get started. Do you want to be lab partners? Okay. So we will flip this here. Okay.
Okay.
Ashley, you want to come sit here? Okay. And then, do you want to work by yourself or do you want to work in a group of 3?
Either way.
Okay. You can work by yourself if you want.
broadcast.
Or you can work with them.
Hello, people
First is fine.
of the internet.
Okay.
Hello, people of the internet. Okay. So,
Let's do this.
do you want to talk first?
All right. So then,
Writing complete.
you guys, while they're starting on that, why don't you guys come on to the
We're reading this. Hold on. Let's see.
So I already have it set up.
So they're gonna do the onion cells while we do the.
Yeah, I'll read it.
A bunch of red onion cells
Okay.
together.
Hello. Everyone says hello. Okay. We're trying to read it while we're doing this.
I know, it's hard. You'll see what I'm doing. I'm like this. I'm like looking around.
See, like, so many
You
people
gotta
are
be
saying
cross-eyed
hello.
or something.
I know.
Yeah. Okay.
We're doing the balloon. So we're working on the balloon experiment and procedure. Put 10 milliliters of sugar.
So put 10 milliliters of the sugar solution in each tube. Each tube.
We will have 3 amounts of sugar.
Yes.
50%, 25, and 10%.
Yes, Evelyn. Yes. Okay, so in each tube. So we need to.
Yes. I'll just do it. Okay. Hopefully it won't spill out.
Okay, so in each tube. Thank you.
Bringing all the solutions over here since we're all going to work here.
Okay. These look so weird.
Okay. We have 3 ounces of sugar. 10
I think cups of water for right now we're doing. Do you want to do the 10? Like
Yeah.
start off low and
Yeah,
then
we will.
And then here you can put a sticky note like in front of it so you can label which one it is.
Okay,
Someone says, I have to go. I'll watch the recording.
Okay.
Oh,
that's
okay.
fine.
That's fine. See you later.
There will be a test.
Don't spill.
Okay.
You think
So
that's enough?
there's, yeah. So let's put that right there.
That's a 10%. And now, what's 4 more? Okay,
Yeah.
this
What
is
do you
the
think? It looks cool. The cells.
Okay.
We have to put it in each tube.
Now we're doing the 25%.
I was like, who's that?
I got scared. I almost dropped this thing. Okay.
Okay.
And then we're
Let's get
doing
it.
the,
No one can see you.
Actually, do you want
There you
to
go.
look at me?
There you go. No one can
we're
see
doing
you.
the 50% now.
We have all the supplies, so we might as well. Right.
Okay.
Do you want to get?
Okay. And then
And then
we're
we-
gonna do the regular water.
Do you want to get 25?
Yeah.
Or excuse
There's
me,
nothing
10?
Anybody have any
So
questions?
you're gonna
What
put it
happens
there.
if we have all the materials except the
with-
balloon?
That's
a good question. What happens if someone has all the materials, well, not all, but except the balloon? Like they have the rest, but not the balloon.
So they can look at the bubbling
Okay.
because the yeast will bubble, right? You can see the bubbles.
Okay.
You could also make bread,
make bread.
Make bread.
and
For
you'll
your
see
family.
cellular respiration, and then get to eat it.
Can they find that online? Like, can they look it up? How do they?
Like examples.
Yeah,
and stuff like
examples. How to
Of
make
how
it.
to make bread.
Yeah.
Yeah,
Okay.
that's
Well,
pretty
just-
easy. It's basically flour, yeast, water, and oil.
Okay.
That'll be good for like.
And it's very therapeutic because you get to pound the dough for like
Mm-hmm.
quite a bit of time.
We should have done
Okay. You're welcome.
that. Okay.
Okay.
So-
So
Which one is the 10%? This one? Label them?
did you label them?
No. Okay. I know this is
Okay.
regular water, and this is a. That's a 25. This was a 10, and this is a 50.
Yeah.
Okay,
So-
so let's get this.
Good call.
Okay. You
Okay,
label them.
make sure to label people.
Yeah.
'cause
Label.
We're doing it wrong right now. Okay,
Okay. So 50%.
you do 50. I'll do 10%.
Okay.
Okay.
So 50 was. This one.
Uh-huh.
I'll put it like
10
that.
is the one next to it.
And then
the
the regular.
water is over here. Just put H2O or whatever.
Okay.
And then the 25.
You got it. It's this one?
Yes.
Okay.
So we just watched them do it. Don't they have to explain what and why they're? Yep. Good question again.
She'll explain to you why.
Okay, first we have these test tubes right here with water. We have 10%, 50%, the regular water right here,
25. Okay, the 25.
and we use, what did we use for the each one of them? Like 10%?
Anyway,
Okay. That's the regular water.
this is a 25. This is the 10. Okay.
Is it tap water or bottled water? It's tap water.
Uh-oh.
water, I think.
Where does this go?
That
Oh
one
no, it's
goes-
10. Okay, good.
Yeah.
Ooh, good thing I
Is it tap water? Tap water.
It's tap water.
Yeah,
So-
tap water.
And then,
I used to.
yeah, it's tap water.
Okay.
And why are we doing this?
To see how the amounts of sugar impact the rate at which yeast ferment.
Okay. Did you hear that? Okay.
Hopefully everybody listens.
Well, no one's like responding, so it's like we have 16 people.
Okay.
Oh, nope. We
A lot
only
of
put 10.
followers. Okay.
Only 10.
Second step
Of the
of the procedure.
50%.
We gotta add a little less, ideally 1/8 or 1/4 teaspoon of yeast.
So
to
we're using
each
this
test tube.
for the yeast, 1/4 right here. Okay.
Now grab your yeast.
Got the
Yeah,
yeast right
we'll do
here,
that part
people.
later. So you guys can go ahead and start
Like
on the
scoop it
cellular
on.
I'll
respiration.
scoop it.
Hopefully.
Is that good?
Wait,
Ah,
but because when we're gonna put it, shouldn't we ooh, how should we
someone
do
said
it?
add me on Snapchat.
Okay. Oh my
I
gosh,
will.
they even put their
We do
yeast
have
in.
a funnel. I don't know if it will help.
Okay.
Let's see.
Um,
It's basically the same exact size as the tube.
Okay, here's the yeast in the. Can you guys see it? Here's the yeast right there, and we are gonna
Okay, you want to put that
to
right
each
here?
tube. Yes. So 1/4 to each tube.
So we have our 10%,
Ooh,
25.
where should we do this over?
plain one.
But since we're gonna put it in, it's
Okay, so
gonna
now
have
I'm
to
just
be
gonna
right,
add
so
Oh,
gonna-
yeah.
Oh,
the yeast.
Oh,
we're gonna
okay.
use
We're
a
gonna
funnel.
take the balloon.
If you don't have that,
You're
just
gonna
get
add
like
the
a
yeast.
Ziploc bag
You're gonna
and
put the balloon right
take a
on.
little side of it. There we go.
Oh, good job.
I know.
That's awesome.
I learned this from YouTube.
Oh, there's Ty. I mean,
Where's Cody?
I don't know.
Classroom run.
What?
Yes.
Anyways,
You want the funnel?
it was a student that came in. So. Okay, we need.
funnel.
Notice how he didn't say, what are you guys doing? He just was like, where's this person? And walked out.
I know.
Just like we're playing with stuff,
Yeah.
going over here.
Okay, hold on. Just keep that there. Okay, now we gotta put the balloon. Balloon in.
Can you help with that?
Yeah.
Don't drop it. No pressure.
Thanks.
Thanks. Okay, hold it tight though.
I'm afraid that thing's gonna break like.
Yeah,
Okay.
it's gonna
So
pop.
now you're just going to put your finger on the
Oh, well.
Okay, is it good enough, guys?
That's
Good.
good.
Oh my gosh.
You might
Okay,
have
each
to
one?
Put it there, I think.
Potentially. Try not to get it in the balloon.
Okay, each one.
Or you're supposed to like shake it up
Yeah.
first a little. Oh, yeah. Shake it up first. Okay.
And note the
Mm.
time, guys. If you're taking like 5 minutes to get the yeast in for the next one, right, that's going to impact it.
12:39.
Okay, I'm
So
shaking
write down right now
Okay.
the time
The
that
yeast.
one's starting.
in the
12:39.
water. This is the regular water.
39. Yes.
Oh my gosh. It won't mix very well.
Yeah, you might have to.
See, I don't want to get it in the balloon.
It's alright. You might have to vigorously shake
Yeah.
it, but just put your finger on the top. The little goes in the balloon. It's okay. Okay. See, there we go. Now the yeast
Are we using your information for the lab to turn in?
Yes.
Yes,
Yes.
you are.
Okay. So that was the first one. So write down the time.
This
It
is
is
like
12:40.
that right there.
We'll shake it better.
So above
Ugh, okay.
this, right next to.
Ew.
I think it's at the
14% there, right?
Is
12:40.
that
That's when
good?
you start.
Don't laugh at
And
me.
that way we know when 5 minutes is done. Alright. Alright. You want to go grab the funnel again?
Oh my goodness.
Alright, my yeast look like they're
Wait, so we
Working
put
already.
no, you put wait. They put our names on it or
no,
their
they
names?
just put
No.
your data.
No, yeah.
You pretty much
They do their own thoughts. They just use your data. So they should have their own hypothesis, their own everything
Okay,
else.
that's
Okay, that's good.
That's
That's good. That looks like oatmeal. I mean
I had oatmeal this morning.
Does it look like that?
Yes.
okay.
Okay.
Sorry.
Let's do it
Do
again.
another one.
This
How many
time
minutes does that have to take?
It's 12:40.
We're right.
Like 2 minutes.
So write down the time that you're starting that one.
I'm gonna be there.
Okay.
12:42.
I need my phone.
So you know.
Alright.
Okay. Regular water. Okay, now this one,
Thank you.
which percent?
You're welcome.
25%, right?
Bye.
Bye.
Yeah, 25%.
Okay. And what time is it? 12:40.
It's so how do we see what they're doing
25%.
and type at the same time? How do they see what we're doing and type at the same time?
Right now they're just watching and asking
Yeah.
questions. That's it.
Okay, just watch and ask questions.
Yeah, that's it.
No, just listen. Just listen. Don't divide the screen. Just listen. Okay, I need the funnel. I need the funnel. Oh, she
Oh, they got it.
okay, you can pour it this time.
Here, you can have it back. Just want to use it one more time. Okay. These two are going to start off the same time.
Oh my goodness. Hello.
Evelyn.
Evelyn.
I thought his name was Ruben or something. Okay.
Let's okay. You're shaking it.
You do have to shake it pretty vigorously.
Yeah,
I
you do.
apologize for not knowing
And
that.
this is the 25%.
Okay, shake it.
It really does look like oatmeal.
He's
So
taking
going for
pictures.
50%.
Oh, this one again? Evelyn.
There's a kid taking pictures. Okay.
Oh, yeah, I should take pictures so I can put them on Facebook.
Oh yeah. Okay, did you shake it really well?
Yes, I did.
Okay, now we're doing the 10%. And they're typing. Okay, they are saying, write out it on a piece of paper. What? Hold on. Hold on. How do you move up on this chat box? I didn't see the.
Good question.
How do you?
This is pretty much me every time like I'm trying to deal with this thing. You just scroll.
Oh, I just okay.
Yeah, and then you can scroll down.
Got it.
Uh-huh.
What time did you
I'll also
put
take a
that
picture
one
of him taking a picture and put it on Facebook.
in? We started at 12:41 and ended at 12:43. And that one, what percent?
This one?
Okay.
The red one. Oh.
Someone said truth or dare. What? We're not playing a game here, people.
This is.
Unlike you guys who are just chilling at home, we're working here.
Yeah.
Are you guys doing it too? Are you gonna do it too?
Why are they playing truth or dare? Why are you guys playing truth or dare?
This is a class.
All right, so you're
Now we're gonna do 10%.
Okay, I can't get out of this thing. This right here. How do you get out of that?
But just click something else. Like
Click.
there we go.
Okay, there we go.
Oh, we have.
One new message. Okay, hold on. We're coming.
Okay.
No, I'm.
Did you just put Indian?
Oh my God.
What? Write it out. Scroll. What are you talking about?
Stop tampering with. Get out. Press X. We know, Evelyn.
Oh my gosh. Okay, so for observation, we should write down what you're doing and later go back to the lab once the session is in. Is that true? So, for observations, they should write down what we're doing.
Mhm.
They should write it down.
For right now, they should just look at what we are doing. Get out of the screen, Evelyn. Center of attention today.
We'll do a little more shaking
Now we're doing the 10%.
on this guy too. That's a little more. Yep, you can.
Okay, we have 16 people.
Let me know.
Cool.
Still got.
Wait, what happened? There were
Tell
18
us
people.
what's happening with the
One
stuff.
stuff. Okay,
So
stuff.
right now it's doing nothing.
Doing nothing.
Yeah.
That's
all I can say. Okay.
Okay, right now,
Oh yeah, that was.
we put the yeast in the regular water,
Which is the yellow.
which is the yellow balloon.
And the 25% is the red balloon.
Yeah. And then we shook it up, and then we put it down so it can chillax. Just chill right there. What will happen if you put it in soda?
I think it will raise up. I don't
That would
know.
be perfect. And then the rest
This
is
kid got a question. How about if
What if you put it in soda?
Yeah.
the yeast.
Yes.
What will happen?
Well, it is water and sugar basically,
Mhm.
right? So it would probably work. That would be an interesting thing to try, comparing water and sugar to soda.
Try it out and let us know.
Yeah, try that. It might work.
Let's go to big.
How are your balloons looking? Ours haven't done anything yet.
Ours either. There's like.
Nothing.
Let's start with
How about vinegar?
vinegar.
Yeah.
I don't think vinegar would work.
Yeah.
Yeast won't ferment in vinegar.
So we'll do nothing.
You really have to shake it, right? Paper towel?
Okay. So
Now
anyways.
we are
gotta
going to
pull
do
them out
the
of there.
10%.
Okay,
almost
so
did that.
get your test tubes made so you guys can come check out the onion cells.
Okay.
Let's do this.
Okay.
Where's the yeast?
Where's right
Oh.
there?
I'm blind. Okay. So this is 10%.
How can we dump it all over?
Be a funny podcast. Put it all over Snapchat.
Put it all.
Don't do it.
No, don't put it on Snapchat.
Yes. So
This is 10%. Okay. Will you shake it this time?
Oh, shake
Okay.
it.
Just gonna shake.
Okay. Okay, is that good?
A little bit more so you won't see the little.
See bubbles in there. working.
Okay. Do you even have a Snapchat?
It could also be that it's not enough solutions since I had to cut it.
Okay. I'm talking about unnecessary things, guys.
Yes.
Like, I wonder if we tried.
Okay,
this.
anyways.
Now
So, what one was
What's the
that?
time for this?
What one was that?
What's
10%?
the time for this one?
Okay, we just shook up the 10%. And the stickies.
I'll
And
probably put it in a couple of days.
Sorry, they're not sticky. There's also tapes.
so this is the 10%, the purple balloon. We just shook it up.
and right now it is doing nothing.
You guys want to see?
Others.
Our balloons are doing absolutely nothing.
Be like a hand model.
And for 20%.
There we go.
For
For 20% off. Okay.
Okay, more people are typing. Okay.
When you shake it, it's probably not
well,
doing anything.
The
since
balloon
balloon
is on when you do it, and
pressure is too much. I don't know though.
I don't know.
I don't know
that.
either.
All right, I'm trying a huge one.
Dang.
Okay, someone says when you shake it, it is probably not doing anything since the balloon is on when you do it.
And the pressure
And the
is
pressure
too
is
much.
too much.
I don't know that.
For the yeast?
Yeah.
That's possible. But we want to trap the oxygen in there. We don't want extra oxygen. But you can try taking the balloon off for a second, shaking it a little bit, and then putting it back on. I mean, you're going to have to put the balloon on.
Okay.
That's
Wait,
not
shake it with my finger.
Yeah. It's just yeast and water. It's not going to hurt you.
Oh,
This
this one's-
also, this takes time, guys. The cells have to produce. Ooh, this one I'm feeling a little air in.
They're just doing a better job than us.
Oh, whoa.
See, that's why I wanted all of us to do it so that we can
Okay.
experience.
Oh my god.
Oh
Oh no. Okay. Wow, your idea. Your idea was
Oh, is it 48? We're supposed to.
Should I
do
put
this
it
one already?
Okay. This is the
Do it right now. All right. Well,
What
we
is
haven't
it?
done anything. So we can just
the yellow is the.
Do I put it back?
Yeah, put it back.
Okay, let's see if this will do it.
That one
This one is 45.
Here we go.
That's it.
Okay.
Google
Google Calendar. You have something that popped. Yeah, it says prevent this page from creating additional dialogues.
Just push OK. I think I have a meeting at 1, but I might have to be late to it.
Okay. How do you?
Just press the little X at the top.
Yes.
Yeah, the red thing.
No.
Oh,
That's
no?
gonna close all
Oh,
the
okay,
tabs.
then you do that. Okay,
there we go.
never mind. Would have gotten.
Guys, I advise you to wash your hands after you touch the yeast because that is gross. It smells.
Okay, so now that you guys almost have all, you got to put a balloon on the water, put yeast in there, and put a balloon on there, and then you guys are gonna come look at the onion sauce.
Okay.
Now this is.
2 people added me. Evelyn, no one cares. Okay, I'm sorry. We're doing a lab.
Where's the funnel thing?
Yeah, why aren't you here, Evelyn? That's a real
Yeah,
question.
Evelyn.
Yeah.
You notice there's no boys here?
It's an all girls.
It's a problem. Actually, it's great.
Yeah.
Girls in science.
Okay. Can you pass me the funnel, please?
Can I have the funnel, please? Okay. So he said, "Ouch." LOL.
Well, it's true. Okay.
From I'm the only boy that cares about my grades.
Oh my goodness. Okay. So this is the 50%.
Is there any boys in this broadcast, anyways? Like,
I don't know. There's a Bruce and then Evelyn.
and then there's
So,
an
in
Evelyn.
the huge one, guys, you can feel
Yeah.
you can feel the air. It's getting in there.
Yeah. The yellow one before I took it off, there was air actually.
Okay,
So,
just a little bit.
yeah, just
Okay,
a little bit.
So
Are you
we'll
shaking
give it some time.
Now we're gonna shake the 50%.
No,
No, that's
you are
more,
not
isn't
the
it?
only boy that
No,
cares
this
about
was a
Oh,
grades. No, you are not the only boy that cares. See, Evelyn, there's another boy that
More
actually cares.
boys.
There's a boy that commented and said no.
He cares about his grades.
Yeah. Okay, that's good.
Okay, well, you two take a pause there.
Okay.
I'm gonna send them pictures of the cells. You're gonna go look at the onion sauce.
Oh, cool.
We're gonna take a break.
We're taking a break from you guys because
Too crazy.
Okay. So they're looking at the onion cells. So I'm just gonna send you guys a link to a Google image of onion cells. It's pretty simple. You could also Google it yourself.
What do you write in here?
You're gonna write the diameter of the balloon. So you might want to like note what time you need to be checking them.
What will happen to the
Let's see. This is a good one.
salt water in March? So what's
All right. So this, when will this be due? Good question. Yeah, ideally today, but I will put it the rest of this week. Okay. Here's a great, whoa, that's a really long link. Can you guys even click on that? Okay. Oh, it just takes you to this, which is not what I wanted, but whatever. That's basically what happens when you Google red onion cell in the microscope. That's what they're looking at over there. So I actually took a razor and I shaved the edge of a red onion, right, and took the really thin slice, put it on a slide, and put it under the microscope. And that's what it looks like. You can see a bunch of individual cells. In some of them you can see the nucleus, but it's not super detailed. Yeah, it's pretty weird. And then we're going to add salt water in a second, and we're going to see how that changes it, which I think I showed you guys before in a previous virtual lesson, but it never hurts to see it multiple times. Meanwhile, the yeast is working away. Ooh, this one's starting to get some air in it. You can see all the bubbling here, right? That's the cellular respiration. The yellow one's getting air.
That's the one that I took tap water.
That's water?
Yeah.
The control is bubbling. That's a problem. Are you guys sure you didn't get them confused?
Yeah.
Okay. I'm a little scared. Yeah, I'll send you the results if you have to leave. I'm going to record this all the way until the end. We still have probably about 15 more minutes of data collection. But if you don't want to see the rest of it, because it's basically going to be us checking it and sitting here, then you're welcome to leave. And I will, in the document, I will update the data that we get. Okay. Did you guys see the onion cells?
Yeah,
Oh, no, right?
We did our prediction.
Oh,
Yeah,
good. Well, look at that. Because I want to put the salt water.
Is that live virtual lesson?
No, there's still a
Hi guys.
They're happy to take a break, probably. They're like too much learning.
Oh, wait a second. There's no regular water.
Yeah.
Okay.
How is it?
That's pretty cool, right?
Yeah. Whoa.
So 1 minute,
All right, how are you doing there, Ashley?
25%.
Yeah.
You feeling any air?
So what is it? What do you think it looks like?
A little bit.
To me it looks like the, you know, like the, like, put it up,
I see bubbling going on.
to the sun. Yeah. It looks like little, I mean, like cell walls.
The water one have air? No, there are water ones producing air.
Where is it?
Which is not good.
This way, or it's going like straight. So let's just try it like.
Okay, so this one is.
Okay, in fact, I'm gonna have to tell my 1 o'clock meeting that I'm gonna be late. Okay, so we're just finishing the experiment. You guys, in fact, I think I might end the virtual session because this is pretty much it. But basically, for the next 10 minutes or so, we're going to be measuring the diameter of our balloons, hoping that they grow. And I will send the results. In fact, I'll just send an email with them. That'll be the easiest way. And then DBAs, yes, except I do have a meeting that I'm supposed to be in right now that I forgot about. So call me and I will leave the meeting to answer if you're doing a DBA. All right, otherwise, I'm going to end this. Thank you guys for participating from home. Say thank you to our wonderful lab partners.
You're welcome.
They're looking at the onion cells right now.
Add us on Snapchat,
The best time to call me would probably be 2. Okay, so I'm ending the meeting. Goodbye, everyone.
What time would you prefer?